So I made the first section of this explanation dedicated to just definition of words. So the full um, document to this is going to be in the notes that's going to be uploaded to the Google Drive, which I'm going to link in the description of this video. Um, you can look over them before we start, and I would advise you to do that in case you're not familiar with um, the neuroplasticity terms that were used in this lecture, because it's going to make everything way easier for you.
So we all know the different parts of the neuron. We know the nucleus, the axon, the myelin sheath, the axon terminal, Schwann cells, nodes of Ranvier, the axon hillock, cell membrane, cell body dendrites, and so on. And there's actually more that are not listed here. Before I move on, actually, I just want to say something small about Schwann cells. This should tell you something. If you want, you can pause the video and guess what I'm about to say. So if you guessed that Schwann cells on a neuron tell you that this neuron is located in the peripheral nervous system, then you would have guessed right, because in the central nervous system, we have oligodendrocytes, which make the myelin sheath. Okay. So moving on. Okay, so what do we have here? What are the ones involved in the formation of new synaptic connections? So when we talk about the different parts of a neuron within the context of neuroplasticity, there's a reason. So within, or when we're talking about neuroplasticity in general, there are two main processes that lead the way, basically. We have neurogenesis and synaptogenesis. Let's talk about synaptogenesis first. So what is synaptogenesis? I'm going to tell you right here now, whenever you see the word genesis at the end of a word, it's going to mean the formation of something new. What is the something? You go back to the first or the prefix of the word. In this case, it's going to be the synapsis. So formation of new connections, which is what synaptic connections are between neurons. New connections between neurons. Okay, sorry for the many abbreviations. So what are the ones involved in the formation of new synaptic connections? What does this question mean? What I'm asking here is, what are the parts of the neuron that are involved in the formation of new connections between neurons? When we have two neurons that form a connection for the first time, what are the different components of the neurons that are involved in this process? So let's look at the ones we have here. Oh, there you go. First of all, we have dendrites, synaptic knobs, axonal branches, and neurotransmitters. Neurotransmitters, again, they're not really a part of the neurons, but just remember them. I don't know what to say. Okay. Okay. Oh, and if you want to look into what each of these are, you can take a look at the index, which again is going to be in the notes, which is going to be in the Google Drive, linked in the description down below. Yeah. Okay, moving on. Okay, so that's it. We just finished the first section. Moving on to the next section, we're going to be talking more in depth about neuroplasticity. So we talked a little bit about synaptogenesis, and we are going to talk about it a little bit more. But before we go into that, let's ask ourselves, what is neuroplasticity and why do we need it, and what does it do for us? So we know that the brain is a dynamic structure, but what does that mean? Dynamic means something that is easily changeable. So what does it mean when we say that the brain is easily changeable? This means that the brain can adapt to different learning experiences and different situations through neurogenesis and synaptogenesis. And this is what neuroplasticity is. It means that the brain is able to increase or decrease the amount of neurons or the amount of synaptic connections in order to adapt to the learning in order to adapt or react to the learning experiences or stimuli. Okay, why do we need neuroplasticity? So, neuroplasticity is actually the reason we can learn and we can remember. So, it's very, very important. It's the main reason we have memory and we can learn. Yeah, that's basically it. So, moving on to the main components of neuroplasticity, or what the processes that give us neuroplasticity, if you will. We have neurogenesis and synaptogenesis. Again, just like I said before, genesis just refers to the formation of something new. What is this something? Refer to the prefix. So it's the formation of new neurons. That easy. And synaptogenesis, just like we mentioned earlier, it's the formation of new synaptic connections. Okay, so let's dive deeper into how synaptogenesis actually happens. So there's three main things, or three main steps that occur before a new connection can happen. Three or two, I mean, depending on how you see it, we'll see. So first of all, you need a repeated long-term effective stimuli, something that is remarkable, that your brain is something that is remarkable enough for your brain to remember and retain. So the first thing you need is effective long-term or repeated, again, stimuli. Once you have that, before you can move on and make this new connection, you're going to need new protein synthesis. Once you have these two, synaptogenesis occurs and you have a new connection that forms. So you could think of it as two steps. Okay, moving on to neurogenesis. Neurogenesis is a bit more complicated, and you would guess so because it's the formation of new neurons, whereas synaptogenesis is just the formation of new connections. So neurogenesis, how it starts, we start from stem cells. We have progenitor stem cells, or they're also called neurogenerator cells, that are stimulated to differentiate into neurons. So how does this happen? How is neurogenesis kick-started? Well, there's a couple of things that we need, or the progenitor stem cells need, in order for neurogenesis to happen. First of all, we need neurotrophic factors. We need cell adhesion molecules. We need certain neurotransmitters like serotonin. We need some hormones. Sometimes, in some states, inflammatory factors kickstart neurogenesis. So inflammatory factors are also things that could activate progenitor stem cells. We also have genetic factors. And lastly, stimulation of mossy fiber neurons in the cerebellum. Okay, so what do each of these mean? Again, they're all listed in the index, but I'm just going to go over them really quickly. Neurotrophic factors are proteins that stimulate growth proteins, and they include growth factors, which we're going to be talking about more in detail later on. They're actually listed in this lecture. We also have cell adhesion molecules, which are exactly what they sound like. They help cells stick together and communicate with each other. Certain neurotransmitters like serotonin, and I'm sure there's more. But the thing is, your professor did not list a lot. So if I don't list a lot of details, it's because it wasn't listed in your lecture, and I don't want to confuse you by adding information that is not going to help. But feel more than free to look these up, because these videos are supposed to be summaries or information banks, if you will, that are going to help you study. But I would honestly encourage you to look it up in your free time. How do these different factors go and influence progenitor stem cells exactly? What's the actual molecular mechanism? I'm sure it's really, really interesting, but I'm just here to help you study for your exams. And yeah, okay. Okay, so besides these, there are also some other things that influence neurogenesis, but influence it negatively. So they inhibit neurogenesis. What are these things? It's things like age. So with age, as you grow older, your neurogenesis rate declines. You also have things like stress, the release of glucocorticoids negatively impact neurogenesis. Also, lesions in the hippocampus. So these are all things that will inhibit neurogenesis. Also, don't worry about my kind of messy notes. I, again, have much more organized notes that I'm basing all of this explanation on that are going to be linked down below. So, we talked about neurogenesis. We talked about what it is, where it starts, and how it happens, and what inhibits it. Now, when you have a progenitor stem cell that is maturing to become a neuron, it needs guidance. It's not an easy journey. So it needs these things called neuronal development or development guidance cues. And these guidance cues are molecules that include netrins, simorphins, slits or SLITS, and ephrins. Again, these were not described in detail in the lecture, so I would assume that you would not need to know the details. But again, in your free time, it would be really interesting if you were to research about them. But these are just things that you are required to know. These are the guidance cues. Cues. Okay, and that's basically it. The last thing we're going to talk about in this section is why can't regeneration occur easily in the CNS? So this is actually a major topic in research, regeneration in the central nervous system. So there's two main reasons why it can't happen. First of all, we know that in the nervous system, we have oligodendrocytes, which their main function is to synthesize the myelin sheath. However, they can inhibit the regeneration of neurons in the central nervous system by releasing inhibitory substances. When and how does this happen? So when a neuron in the central nervous system is injured, these oligodendrocytes release inhibitory substances like NOGO proteins and MAG proteins that inhibit any potential regeneration. This is one of the main reasons why regeneration or yeah, regeneration is very difficult to happen and very rare in the central nervous system. Another reason is because of astrocytes. And you might be thinking, why? Astrocytes are supportive cells. Well, astrocytes actually form scar tissue right after neuronal injury. And this scar tissue acts as a physical barrier that stops any regeneration from happening. These are the two main reasons why regeneration is very difficult for the central nervous system. So in the peripheral nervous system, regeneration is actually kind of encouraged because again, like we mentioned earlier, in the peripheral nervous system, Schwann cells are what make or synthesize the myelin sheath. And these Schwann cells do not release the no-go proteins and the MAC proteins and those inhibitory substances. In fact, they actually release promoting factors. So instead of inhibitory substances, they release promoting factors. Again, these promoting factors were not specified, but if you do want to look them up on your own time, feel free, because it's a very interesting subject. And this is why, so you could say the absence of oligodendrocytes in the peripheral nervous system is what contributes to its ability to regenerate. Yeah, that's basically section 3. So that's basically what you're expected to know of neuroplasticity. So moving on to section 4, which is learning and memory. We're going to be talking about a few things here. First of all, we're going to be talking about the definitions of learning and memory. Then we're going to be talking about the types of memory. We're also going to be talking about where each type of memory is stored or where it happens. And we're going to be talking about what mechanisms each type of memory uses. Okay, so let's start with definitions first. What is memory? What is memory? Memory can be defined as the ability to store learned knowledge in any area of the central nervous system and recall them whenever is necessary. Learning can be defined as the ability to change the behaviors depending on experiences, which occurs with repeating or impressive or remarkable stimuli. You could also recall that synaptogenesis, this is basically what synaptogenesis is. So this is why synaptogenesis is major and why neuroplasticity is very important for learning and memory. So moving on, again, the definitions of each of these are in the notes. Okay, so when we talk about the types of memory, there's two main things that we need to be aware of. First of all, we need to know where each type of memory is stored, and we need to know which set of mechanisms occur with each type, because each of these types use different mechanisms. For example, first of all, your working or your immediate memory is stored in the or not stored, since it's immediate, you could say, stored is technically the right word, but yeah, okay, let's not get into that. Prefrontal area. Your short-term memory is stored in your hippocampus and medial temporal lobe. And your long-term memory is stored in your neocortex. So this is the first criteria that we need to be aware of. Where are they stored? The second criteria that we need to be aware of when it comes to types of memory is their mechanisms. So working or immediate memory uses reverberating circuit mechanism. The short-term memory uses two mechanisms: presynaptic facilitation and post-tetanic potentiation, which we're going to get into in a bit. And finally, your long-term memory uses long-term potentiation. And this is basically what we need to know about each type of memory. So now the last thing that we need to know about this section is how each mechanism works. And this is actually a pretty, not complicated, but this is pretty important. So reverberating circuit mechanism, which occurs again in the prefrontal area, is a neural circuit mechanism that involves a loop of interconnected neurons where signals travel in a continuous circular path. So this is the definition of what a reverberating circuit mechanism is. And I feel like it's a bit self-explanatory. Again, there's no like any crazy details that were mentioned in this lecture that you are required to know, but feel free to look it up in your own time. However, for presynaptic facilitation and post-tetanic potentiation, you are required to know them in a bit more detailed way. So let's start with presynaptic facilitation, which again, let's review, is a mechanism of short-term memory. And short-term memory occurs in the hippocampus and medial frontal lobe. Oh, sorry, medial temporal lobe. My bad. Medial frontal lobe. No, medial temporal lobe. Okay. So, in order to understand presynaptic facilitation, we need to review how normal signal transmission happens. So basically, on a very basic level, in very simple terms, for a signal to be transmitted from one neuron to another, the presynaptic neuron needs to be depolarized. Depolarization needs to occur, meaning it needs to lose its negative charge. And that happens when calcium rushes into it, because calcium has positive charge. When depolarization occurs, this is when the signal is transmitted. Now, when this happens, this mechanism, or you could say the neuron, the presynaptic neuron, also has a built-in mechanism where repolarization immediately occurs to ensure that the postsynaptic neuron doesn't get activated over and over again. So it kind of puts like a limit to how much of the signal can be transmitted. So this repolarization mechanism ensures that the neuron does not get overexcited and limits signal transmission. So with presynaptic facilitation, what happens is a third neuron comes into play, and we call this the facilitator, and it causes delayed repolarization, which allows longer duration of signal transmission. And how that exactly happens is what we're going to get into right now. Okay, so I explained a little bit about how action potential normally carries out. I think I'm going to re-explain it just really quickly on a molecular level, just so we're on the same page when I start explaining presynaptic facilitation. So normally, when a neuron is in a resting state, it likes to stay at a negative charge. And when an action potential happens, it needs to disrupt this resting state by depolarization. How does depolarization occur? By the calcium channels letting in calcium ions. This adds positive charges. The neuron is depolarized. Once depolarization occurs, the vesicles fuse with the surface and the neurotransmitters are let out into the synaptic cleft and they bind to the synaptic knobs. So when this happens right away, there's a built-in mechanism, just like I mentioned earlier, where potassium channels open and they let out potassium ions in order to restore or repolarize the neuron, to restore it to its resting state so that the postsynaptic neuron is not constantly being activated. So kind of to control the transmission or control the action potential a little bit. So so this is what normally happens. When we talk about presynaptic facilitation, the main aim is to delay this potassium channel opening a little bit in order to provide a prolonged signal is basically the main objective. So how does this happen? First of all, we have something called a facilitator, which is just a third neuron that comes in and provides the presynaptic neuron with serotonin. Now, that's all this neuron does. We get the serotonin, and this serotonin binds to the metabotropic receptor. And this is where this cascade of this mechanism actually starts. So the metatropic receptor activates G protein. G protein activates adenylate cyclase. Adenylate cyclase activates the synthesis of cyclic AMP from ATP, or the conversion into cyclic AMP. Now, I'm not sure if you're supposed to know all of these steps. It is more preferable that you know these components, but the main objective of all of this, the main objective of the serotonin binding to the metatropic receptor and initiating all of this, is that so we can get a protein kinase C, so we can activate the protein kinase C. Now, why do we need to activate this protein kinase C? What does it do? This protein kinase C, once it's activated, it starts phosphorylating the potassium channel. When this potassium channel is phosphorylated, it's like you're not impairing it, but you're slowing it down. When you slow down the potassium channel, these potassiums that normally go out of the cell and cause repolarization to happen, this is slowed down and you have a prolonged depolarized state. When you have a prolonged depolarized state, this allows the action potential to keep on fulfilling itself, if you will, or basically allowing the signal transmission to keep getting transduced just a little bit longer. This is basically what presynaptic facilitation is. Again, recapping, this is a mechanism of short-term memory, and which happens in the hippocampus and medial temporal lobe. All right, so that was the first mechanism that is used by short-term memory. The second thing we're going to be talking about that is also used by short-term memory is post-tetanic potentiation. This one's a little easier to explain because it doesn't have any crazy mechanisms that you're supposed to know. All you have to know is that it's a short-term boost in the strength of communication. So when you have two neurons that are communicating, and they have basically rapid and intense, there's a lot of neuronal activity happening here, lots of intense activity, maybe a new memories being formed, for example, like in short-term memory, which is where this happens. This also happens, I'm pretty sure, I don't want to make stuff up, but let's just stick to what I know. So post-tetanic potentiation is a boost of the strength. So you could think of it as like a super boost, kind of like those mushrooms from Mario, where they get like a boost of energy. And that's it. That's literally just what post-tetanic potentiation is. Again, short-term memory in the hippocampus. I just like revising these every chance I get because then I'm never going to forget them. This is what I do with everything. Again, hippocampus and medial temporal lobe. That's it. That's basically it for... So we just finished short-term memory, working immediate memory. Now we're moving on to long-term memory. So we know that long-term memory stored in the neocortex and utilizes long-term potentiation. So what is long-term potentiation? Let's get into that right now. Long-term potentiation. Okay, so long-term potentiation. This is the last mechanism involved with memory, and it's involved with long-term memory. Again, long-term memory, where does it happen? In the neocortex. Just a little revision. Okay, so I have a little summary over here that you can pause and read if you want. I can also add it to the notes, but it's very simple. I took some of this text that your professor provided you with, and it's very complicated, and it's not fun to read it all, but I summarized just a small part of it that talks about long-term potentiation, and this is what I gathered. So let's just read it a bit and then break it down. In simple terms, when there's long-term depolarization in a nerve cell, it causes more calcium channels to open, leading to increased release of neurotransmitters. So when you have long-term potentiation, the neuron is encouraged to let in more calcium. You have prolonged depolarization. And this leads to the release of more neurotransmitters, which is easy to gather since we already know how this normally happens. Okay, so when you have... So let's continue. This prolonged activity triggers the synthesis of certain proteins through a process involving gene expression. So this is basically it, just going into more detail. So when you have more calcium going in and you have more signals being transduced, you are strengthening the synaptic connection. You have a really strong connection here. And this connection is strengthened even more when we have glutamate, which is one of the neurotransmitters, binding to the AMDA receptor. Okay, and this binding triggers the NMDA receptor. And this NMDA receptor stimulates even more calcium to go and depolarize the postsynaptic cell. And what this does is that it strengthens the synaptic connection even more. So it doesn't stop there. These calcium ions, they actually have two main functions. So the calcium ions that are stimulated by the NMDA receptors, not only do they increase or prolong depolarization, they also activate proteins like PKC, PKA, and MAPK. Activate PKC, PKA, and MAPK, or MAPK. Actually, I should put this on the side. Here we go. And what these do is that they, again, help strengthen the connections between the cells even more, which enhances memory by lowering the threshold for activation in the new receiving cell. So the activation of PKC, PKA, and MAPK lower the threshold. They lower the amount of energy that is needed for this postsynaptic cell in order for the action potential to follow through, basically. So making it easier for you to recall that memory. So if you've ever forgotten something, it's probably because you haven't used the synaptic connection in a really long time. And this is how that happens. So, in simple terms, when nerve cell connections aren't used for a long time, they become weaker. So, this is a really recurring theme: weaker and stronger. When you have stronger synaptic connections, they're more likely to be stored in your long term memory. When your synaptic connections become weaker, they're at risk of becoming depressed and then you losing that memory. So if their frequency is lower than 10 to 20 hertz or 10 to 20 times per second, it leads to less calcium entering the cell. So if you have lower than 10 hertz, this causes less calcium to be let in the cell. So when you don't have any calcium going into your presynaptic neuron, depolarization cannot occur and it stays in its resting negative state. This deactivates or turns off the connection instead of strengthening it. This is pretty common sense if you think about everything we know up until now about how neurons communicate. Specifically, important proteins like CAMK2 and PKC are inactivated and receptors on the cell surface are taken inside the cell. This process causes the breakdown of proteins, making the connection weaker and less effective. So these are the two main proteins that we need to keep in mind that are deactivated: CAMK2, PKC. They are deactivated and the receptors are broken down via endocytosis. They're taken inside the cell and then broken down. And this is how we lose a memory. Okay, so the last thing we're going to be talking about is epigenetics. Epigenetics, its basic definition is it is the mechanism that is responsible, or it refers to the set of mechanisms that are responsible for regulating gene activity without actually interfering with DNA sequence. So you have a gene over here. We have a bunch of genes, actually.
Mhm.
Epigenetics is when you go in and you alter. So you have genotype and you have phenotype. Genotype is your genetic makeup, and phenotype is how they are expressed, whether that's physically or in your body and how your body works. Okay? So epigenetics is the set of mechanism that goes in and it alters how this genotype is perceived to be a phenotype without actually altering the sequence, without changing up anything. And so basically you could say that these genes are either turned on or off by certain actions, but the DNA sequence is not affected whatsoever. So what are the mechanisms that are included here that fall under epigenetics? We have DNA methylation, which is adding a methyl group to DNA. Again, these are not gone into detail in the lecture, so I don't think you're supposed to know it, but in your free time, absolutely go in and research about them. Chromatin remodeling, and I feel like most of these are pretty self-explanatory. Histone modifications, and non-coding RNA mechanisms. I'll repeat this again. All these are defined in the index that I've provided in the notes: DNA methylation, chromatin remodeling, histone modifications, and non-coding RNA mechanisms. Those are all defined in the notes that I'm going to upload to the Google Drive, which is going to be linked in the description down below. And yeah, that was it for neuroplasticity. Good luck with your exams.